you are listening to EcoJustice Radio on KPFK Los Angeles, a project of SoCal 350 Climate Action. Our show presents environmental and climate stories from a social justice frame, featuring voices not necessarily heard on mainstream media. Welcome. I am Jessica Aldridge. On today's show, Renewing the Environment with Mycoremediation, host Carrie Kim interviews Danielle Stevenson, applied mycologist and founder of DIY Fungi. She offers education, consulting, and mushroom cultures for people and communities to grow mushrooms for food and medicine and integrate fungi into food systems, mycoremediation, and waste management. Danielle Stevenson is currently a PhD student in environmental toxicology at the University of California, Riverside, where she studies mycorrhizal fungi in soil remediation and sustainable agriculture. She is also founder and advisor to the Healing City Soils Project, a Future Leaders Fellow with the Foundation for Food and Agriculture, and a board member with CoRenewal and the Association for Women in Science in Riverside. Thank you for tuning into Renewing the Environment with Mycoremediation and host Carrie Kim.
Aloha, my name is Carrie Kim, and today we're thrilled to welcome applied mycologist and founder of DIY Fungi, Danielle Stevenson, on the show to discuss mycoremediation with us. Welcome, Danielle.
Hi, it's an honor to be here.
Great to have you here. I want to give a little intro. We live in an increasingly toxic chemical soup, as I think most of us know. At least 85,000 chemicals and more permeate our daily lives, and they are found in our cosmetics, our clothing, furniture, electronics, cleaning products, gasoline, and construction materials, from industrial farming, extractive and pharmaceutical industries, nuclear energy. So all of these chemicals have had great devastating impacts upon our air, soil, and water. And our health outcomes reflect that, including our response to COVID-19, the rise of autism in children, reproductive health in adults, neurodegenerative diseases in the elderly, and cancer in all ages. These are urgent indicators that we must change course. And we are on some levels. So enter the incredible potential of fungi and mycoremediation for ecological restoration. And this is where Danielle comes in. We thank you so much for sharing your wisdom and your knowledge. We want to thank the Tongva ancestors from where our show comes. And let's begin. I wanted to first, thank you for coming on the show to tell us and educate us about this potential of mycoremediation and ecological remediation. Sam Shoemaker of Myco Farm, who introduced us, told me you were passionate and intrigued about mycoremediation, but that you found so few people doing the actual work, you had to actually take it up yourself. Is that true?
That's true, I guess. I actually grew up in a very polluted area, one of the cancer alleys of the world. And so from a very early age, I saw the link, as you have outlined, between environmental pollution and health issues and adverse health outcomes. And so at the same time, I saw the way that nature comes in to help. And I saw, as a youth, I would go on long walks and see shaggy mane mushrooms pushing up through asphalt. And I saw that at an oil refinery near where I lived. I saw, sumac and willow plants coming in also in these really polluted landscapes. I saw algae coming in polluted water systems and cattail. So I was always very driven to do this work and to support the work of fungi and plants and microbes and people who are bringing about ecological healing. And so, but you're right, when, I was doing a lot of community work, actually with plants and, compost, but I didn't know
Mhm.
anything. I didn't really know about mushrooms. And I
Mhm.
wanted to, I knew that fungi played this really important role in soil health.
Mhm.
And, I heard along with, many other folks in North America, what was it, a long time ago, there was that TED Talk by Paul Stamets on
Awesome.
6 ways mushrooms to save the world. And so I thought, hey, I really need to learn more about
Mhm.
fungi. And it's true. I quickly learned that to work with fungi for remediation, one needs to understand their life cycle and how to support them and
Mhm.
what they need to do that work.
Mhm.
So that's how I got into working with fungi. I started growing mycelium, the living part of the organism. And, just from there, that was maybe 11 years ago. I just started trying out everything. I started trying to identify toxic waste streams that I was connected to. And I
Mhm.
can talk more about that
Mhm.
journey. But yeah, it really started with curiosity and care and approaching the fungal kingdom, I suppose, as allies in and just
Mhm.
curiously, humbly, respectfully, um, opening to trying to learn,
And
how to work with them.
now you are getting your PhD in environmental toxicology at UC Riverside,
That's right.
right? And you are
Yeah.
specializing, is it there that you are specializing in soil health with using mycoremediation? Is that a specialty or just your focus?
Yeah, it's the focus of my research, and it
Mhm.
is a bit of a different thing in the department, I think.
Uh-huh. Are you
But
pioneering a way? Probably there's no way really paved before, yeah? Is it?
no, yeah. So I, yeah, so it's exciting in that sense. And sometimes I'm really trying to create space for this sort of work to happen
I think it
as
will
I do.
only grow, and that's the great thing. How do you define mycoremediation, and then in what situations are you seeing it currently being applied?
So I define mycoremediation as working with fungi to clean up or remediate contaminated or polluted soil, water,
Mhm.
and and to regenerate healthy ecosystems. But remediation specifically, I mean, the word means to resolve or correct. And so when we talk about environmental remediation, it's about resolving contamination and toxicity in the environment, in the different environmental media, soil, air, water, and so on. And so myco means, fungal. So
Mhm.
mycoremediation is working with fungi to do that.
And what
Um,
are some of the situations? I mean, I think most people are familiar with the possibility for oil spills. I don't know how much it's actually being applied for that, but
mhm. Yeah,
pesticides, I mean, heavy metals. Tell us what are some of the arenas you're working with, or
sure. So there's different arenas, as you note. So there's the potential to work with fungi and other organisms to clean up after different types of releases, like an oil spill. And there have been a few instances in which that kind of cleanup was tried. I can speak
Mhm.
more about that. But there's also potential to work with fungi to restore and remediate soil. In addition, and to filter water as well. There's some neat work happening with water filtration as well. But the area that I work on, I guess I've worked in each of these areas a little bit,
Mhm.
but an area that I see a lot of potential as well is to work with fungi to deal with really common toxic waste streams. And I love and appreciate, Gary, that you brought that up right at the beginning of the show, because it's not only that we have sort of toxic burden, in the
Mhm.
land, this buildup of
In
metals,
our Mhm. It's in our
right?
bodies.
Yeah, in our bodies, in our water bodies, in the air, but also, our homes are built out of toxic materials. A lot of the things we use in our daily lives may contain toxic components or contribute to the overall toxic burden on
Right.
on the earth. So I think that is an area where there's a lot of potential. Some of the other areas of, soil remediation on a larger scale, water cleanup, and, cleanup after some kind of disaster, those can be pretty daunting and require a lot of resources and organization and skill as well. But the area I see a lot of potential, and an area where I've worked and taught quite a lot, is around working with fungi to deal with, common toxic wastes so that they're
So,
not ending up in the landfill or,
mhm.
polluting our watersheds and
And
things like that.
what would those be? Can you give some examples? What
Yeah.
would be
So
what you consider?
So everything from cigarette butts to dog and cat poop to,
Mhm.
I worked on a project remediating Teflon-based bike lubricants. So
Teflon based, what did you
uh,
say?
bicycle lubricants.
Oh, bicycle lubricants.
Yeah.
Okay. Yep.
So
A
things
lot of
like
hidden.
that.
Mhm.
Yeah,
Yeah, you
exactly.
showed us a picture, or we have a picture of you doing remediation on a bike rag.
Yeah.
Yeah. I was wondering
Yeah.
what that was.
And-
So,
Yeah, and that project, I was inspired by Jay Schindler of Fungi for the People. And I was getting my bike fixed at my community bike shop one day, and I asked them, like, what do you do with your bench tops and bike rakes? Because, like, what is this what is this lubricant stuff?
sure.
And so
Mhm.
they I just remember the guy turning red in the face and being like, man, I mean, we just
Throw
send it to the
them.
landfill. And he took me out back, and there were, just from that week, like a massive pile of garbage bags filled with this stuff. And I did some research
Wow.
into those lubricants. And I don't know if folks are familiar with different PFAS. They're one of these forever chemicals. It's a
Mhm.
class of chemicals that Teflon is one of them, but they're just everywhere, like all in our bodies, in water and soil. And so they're just in so many products that we use. They're in firefighting foams too. So um,
Mhm.
you know,
Fire, yeah, flame retardants and
exactly. So anyways, I thought, wow,
what
this is a problem, contaminant that I'm connected to. And so I started working on that project, working with decomposer fungi and worms, actually, to in this kind
worms.
of multi-step system, really simple system, growing oyster mushroom mycelium on the cardboard from the bike boxes at the shop,
Mhm.
and basically layering in these Teflon soaked bike rags. And then once these decomposer fungi ate through all of that material, then I put the
Amazing.
whole thing in a worm bin, and the worms continue the physical and chemical breakdown of,
Uh-huh.
and then
So
you
that
have
was
soil.
necessary to kind of finish the process. The worms were necessary for that,
Exactly.
to go to completion. So could you give some more examples of the methodology? Like, I know there's different methodologies that are used with mycoremediation. If you could explain some of the different mechanisms or the processes, since we're talking about something natural.
Hmm. Absolutely. So I would break mycoremediation up into different categories, and there's sort of different methods within each. Some of the methods, I will say, are still being figured out. And so,
Sure.
there's a lot of promise that's been shown in the lab, like
Mhm.
on a small scale in these super controlled systems. But then there's actually been very few real world,
Applications.
applications where this has been tried out. And actually, some of them, the few real world applications of mycoremediation didn't fare so well. Lots of really good learning. But for me, I guess I'll go back to the basics. A really simple thing to always consider when you're working with fungi is that you need to support them and provide for their basic needs. So that's going to be, food, whatever it is that they would normally eat in nature. The food needs to be moist. They need to have water so that they can grow and thrive. And typically, there needs to be some kind of air exchange. And so I guess that's a basic principle of mycoremediation. You're going to need to provide the fungus with what it needs to live and thrive. And that kind of leads into something that I've learned over the years of working in this space, is that actually often there are already fungi that have showed up on these contaminated materials, in these contaminated soils, in the waters, even and wetlands, to do the work. And so this is where a best practice is to actually go out to
A toxic site.
yeah. I mean, and that, I think
Your hazmat suit. Yeah.
one of my pictures. Well, I guess, and so if you're not able to collect fungi from the site that you're working on, you can also do something where you'll put out, so say that there's a type of contaminant in the soil on a site you're working with, or a type of contaminated waste material, you can actually put that out in the environment, like in an open but contained dish, and actually see who shows up. So some kind of microbes will probably show up on there, and those are going to be going to be the best ones because they want to do it. They're adapted to that toxicity. They see it as food in some way, and they're adapted to the conditions. And so some of the sort of field trials that have happened with mycoremediation, the reason they didn't work is because a fungus was put in a situation where it couldn't thrive, right? It didn't have what it needed. And so actually, I was catching up with another mycologist colleague who does mycoremediation, and he updated me on the status of a study he had done at a landfill using decomposer fungi with the hopes of remediating the soil there. And so for anyone who's worked on mycoremediation projects on the land, one of the limitations is that it requires so many resources. So you have to get their food. So if you want them to eat and digest, diesel oil or whatever the contaminant is, the organic contaminant, then you're going to have to probably provide a bit of easier to digest food like wood chips. You're going to need to keep the whole thing moist so that the fungus can stay alive, and you're going to have to make sure that the contaminated material is in contact with the living mycelium. And so it's a lot. I mean, if you look at the study done in Washington that Paul Stamets was part of, where diesel contaminated soil from the Washington State Transportation Department was remediated with oyster mushroom mycelium. If you look in the details, you'll see that they used an inoculation rate of 30%, and their piles that they were remediating of soil were 10 yards. So that means 3 yards of mycelium. That's a lot of mycelium, right? So I'm just saying, a lot, when you use an approach where you're bringing a species in that doesn't have an easy time growing there, it takes a lot of resources and a lot of care. Whereas when you use, I don't want to say that, when you amplify fungi and other organisms that might already be present and are adapted to a particular climate, a particular site, it tends to work better, and it tends to also be easier on the remediator, less resources needed. And so just to follow up, so this
I'm going to interrupt you for a second, take a quick break, and we will be right back on that.
Hey listeners, quick break here. We hope that you're enjoying Eco-Justice Radio and KPFK Los Angeles. You are listening to Renewing the Environment with Mycoremediation with host Carrie Kim and guest Danielle Stevenson of DIY Fungi.
So if you could continue, Danielle, on what you were saying.
Yeah, so then to go back to that study done by my colleague at a landfill, he put a
Only
lot
had a
of
small amount of resources.
resources and time. No, it was just a lot of work to set up the study with, wood chips and inoculate with a huge amount of this, decomposer fungal mycelium. And he
Mhm,
had a control which was not treated with the fungal mycelium, just wood
mhm.
chips. And
Okay.
would you guess would that actually at the end of the study they found out that the just the wood chips
Wood chips
alone,
did it.
yeah, they did it.
They
The
brought the
other
much.
yeah, just the wood chips. So what would have happened, I would guess, is that the wood chips stimulated native microbial populations already growing in the soil in that landfill. And so just the addition of like carbon food for them gave them the boost they needed to do their work. And whereas introducing this kind of fungus not from there, it was really hard
Mhm,
to
mhm.
do, and then it didn't
'Cause
actually grow. And then because it didn't grow, it didn't you know like the way that mycoremediation of organic contaminants with decomposer fungi works is through their enzymes. So they
mhm,
actually put out these digestive enzymes because they eat trees and like they eat
mhm,
dead
mhm.
plants in nature. And so a lot of organic contaminants are actually based, were manufactured or derived from something that was alive at one time, right? So they're
Right.
fossil fuel based. And so that's how mycoremediation of that sort works, is through these enzymes, these digestive
Mhm.
enzymes of these decomposer fungi. So if they're not alive, if they don't grow, then they're not putting out those enzymes. And
Got
so
it.
that actually leads, so some folks are working on looking into like, can we just grow the fungal mycelium, in more controlled
Mhm.
environments where it's easier to do that, and then just squeeze out, harvest the enzymes and apply those directly
That's
without.
big. Mhm.
Yeah, that's being
That's
explored. Yeah. So in terms of methods, I'm just sharing some of these. So in terms of you got a bunch of contaminated soil. We've learned a lot through trying to implement, these mycoremediations studies on real contaminated sites with decomposer fungal mycelium that aren't, naturally growing there. There's a lot of limitations and challenges there. Where it's a lot easier to work with those decomposer fungi is in contained systems
mhm.
with toxic waste. And so there's really cool and inspiring work being done around this. There's actually work being done in Mexico where diapers. So
Oh
I
god.
just encourage anyone listening. I get way into waste. You can actually learn about how much like waste is produced in where you live and all the different categories and how many. It's pretty wild, the contribution of plastic or like disposable diaper waste. So these researchers came up with this system of actually like growing edible mushrooms
Mhm.
on disposable diaper waste that they
Uh-huh.
mixed in a bit
Wood chips. No.
with waste with corn stalks, with
Mhm.
agricultural waste.
Mhm.
So they were able to grow these nutritious protein rich mushrooms totally safely, free from contaminants, on diapers and and mixed in with some corn waste. So I guess, yeah, I'm inspired by those. So if you're wanting to work, it's kind of easier in some ways to work with some of those really aggressive decomposer mushrooms like the
Mhm.
oyster mushroom. But I really recommend working with mushrooms like that in closed systems. And any kind of waste is a good one to start with.
But so the translation from the lab or a controlled system out into, the actual toxic site where it might be is just a matter of the resources and not being able to have the right food scenarios. I mean, I'm trying to grasp exactly why it doesn't translate effectively. And I know that there's a big
Mhm.
issue, right, in scaling up, that this is another one of the issues with microremediation. Just it's such a new field as well. There's not a lot of precedent setting that's been done.
Right. So part of it, you're right, is the resources and just the resources to scale up. There's another example actually in California where Fungi Farm collaborated with, I think it was the Klamath River
Mhm,
watershed. And there had been, I think again, it was diesel. I think there was a diesel spill along the banks of the Klamath River.
mhm.
And so they worked on a project. I forget, it was quite a lot of soil that was contaminated. And they basically grew oyster mushroom mycelium because this is one that's known to gobble up diesel. They inoculated like these big rolls of burlap, and then they did these layers of this myceliated burlap with the contaminated soil and added extra straw. So I'll just note, you'll notice that with all of these examples, there's always an addition of an easier to eat food source. So some kind of organic matter. So I'm just noting that because it's, yeah, usually even aggressive decomposers aren't really ready to eat 100%, diesel oil right away. They need a little bit of
Yeah,
something easier to eat
yeah.
first. But then the second thing I'll note again, and it goes all back to this basic need to care for the basic needs of these fungi that we're working with. So they had made these massive piles, these tall piles of soil, and actually these decomposer fungi need oxygen just like us. And they in piles that are too tall, like more than 3 feet, you actually get anaerobic
Uh-huh.
pockets all in the center. So again, the mycelium didn't grow in a big part of it, and so then the remediation wasn't super effective throughout the whole piles of soil. So good learning. So yeah, you're right. It's sort of a matter of resources. It takes a lot to provide the water and get that enough mycelium. But then it's also a matter of adaptation. So there are fungi that don't have those same needs that might be better suited. But the field is, I think it's easiest to grow oyster mushrooms. And so it makes
Uh-huh.
sense to me that when people get excited
Right.
about mycoremediation, they're like, okay, we're gonna use oyster mushroom because it's so easy to grow. And
Uh-huh.
when actually there's a whole range of other, there's fungi. I don't know if you heard, some students at Yale learned about this type of fungus that can actually digest polyester polyurethane plastic as its
Wow.
only food source in
Amazing.
anaerobic conditions.
Wow.
Um, and it was isolated from a plant. So it's an endophytic fungus that lives inside of plants. That's just one example. There's, and what's, and it excites me. I think sometimes people feel a little bummed when if like oyster mushrooms you know
Digest everything
can't
they
cut
want.
it everything in every context. But for me it's actually really exciting because there's actually like really common soil molds and yeasts
Mhm.
that are super effective
Mhm.
at remediating, at breaking down everything from plastic to different types of polycyclic aromatic hydrocarbons, that come from,
Mhm.
like exhaust and
Mhm.
fire and things like that. So processes like that are very accessible to many of us, like really good hot composting practices, things like that will
Mhm.
actually um draw draw in the fungi. They might not be the charismatic like mushroom forming fungi, but
Mhm.
they may be totally
Still in the kingdom.
invisible to the naked eye. Yeah, but they're showing up and they're super hardy and can live in all sorts of environments very easily without like reliable water and so on. And so you'll notice again, this ties back to the story about the landfill and how the wood chips were effective, right? So yeah, composting, adding organic matter, some of these way easier, less resource intensive and
Mhm.
more accessible practices keep proving to be actually very effective
So do you see
remediation
more
practices.
promise in these soil molds and yeast for scaling certain projects? I mean,
Yeah. Yes. Yeah, and it's neat because there's more studies coming out now. I mean, some of the work that I'm doing as well, and have some really inspiring colleagues working on, I mean, are finding, so people are starting to go and actually like we with the personal protective gear and the
Hmm.
and that kind of thing, are going out to super fun sites, so like the most highly contaminated sites and different types of brownfields, other contaminated sites, and actually identifying the fungi that are growing in the soil and growing, helping plants grow on the site and things like that, and sequencing them or just identifying them. And it turns out that a lot of times it's not fungi from the Basidiomycota, like the mushroom forming fungi that show up.
Hmm.
It's from the Ascomycota, which is that's the that's where the yeasts and molds are
Hmm.
found. So yeah, it does, show a lot of promise. Those really common actually soil molds and yeasts do show a lot of promise. A lot easier to scale if you can like
Right.
add, if you, maybe you don't even need to add them. Maybe you can just, add water and organic matter, and it'll encourage their growth and help them do what they're doing.
Hmm. Yeah, it seems like maybe there'll be a way that you can inoculate a site, right? The whole thing with these
Exactly.
molds and yeasts, and that would be a lot less resource intensive, like you say.
Yeah,
Just like someone
exactly.
would spread a compost tea if it could come down to something that simple, you know?
Exactly, exactly. And at some point, I'd love to talk to you about another common contaminant that's treated differently, which is, elemental contaminants, so metals.
Mhm, heavy metals.
Heavy metals, yeah, because as
Yes,
folks may
please.
know, sorry?
Yo, yes, please, I say
Oh,
because we
yes,
need it.
please.
Yes.
Right? So the problem with metals is that they don't break down. They just accumulate. And so we have metals like lead, arsenic, cadmium, and others in our soils building up. They can get into groundwater and surface water. They can get up into the crops, the plants that we eat, into other foods that we eat, like seafood and milk and, and all sorts of foods that we eat. So yeah, they don't break down. And so, organic contaminants are the ones we've been talking about so far. Metals need different approaches. So with metals, you're either trying to stabilize them in
Mhm,
soil so that they're
mhm,
not getting up into our crops, and they're not moving into our water, or you're trying to extract them so that they can be actually contained. And
mhm,
so fungi can really help with this as well. There are the fungi I study in my PhD right now are the mycorrhizal, so the plant friend fungi, the symbionts that connect to the roots of plants and help plants grow. Most all plants on earth have or form these mycorrhizal relationships.
mhm.
And these mycorrhizae can actually help act as bouncers, I guess you could say. Like, so they help plants take up the nutrients they need, but they'll block. They seem
Protectors,
to block and protect, lead and toxic metals from getting up into plants. So that's
almost like
really good.
like our blood-brain barrier for a plant.
Yeah, exactly. And so that's really good. That's really what we want in our agricultural context. But it's really neat and complex because in phytoremediation scenarios, so we're working with different plants that are great at sucking up toxic metals better, way more so than most plants, into their
Mhm,
bodies, where they can
mhm,
then be removed, and essentially big tracts of land can be cleaned of metals this way. So in those contexts, often the soils are so contaminated and so bad, they're harsh soils, that it's
mhm,
hard for plants to thrive. But these mycorrhizal fungi can help plants survive and actually extract more metals. So it's
mhm.
really
Uh,
interesting.
yeah,
Um,
makes sense.
yeah. And then there's other
Wonderful.
fungi that actually are commercially used in wastewater treatment,
Mhm,
and different types of water systems, because fungal mycelium is generally pretty good at sort of absorbing metals. So and this is what's neat in this context, this seems to occur whether they're dead or alive.
That's
And there's,
interesting.
yeah. So fungi, many different types of fungi can help in the different contexts where we're needing to either stabilize metals or extract them.
So to stabilize them, though, is there some kind of maintenance that needs to be done to ensure that that continues over time?
That's not actually known. And so actually that is basically one of the research questions of my PhD is I'm doing these experiments in greenhouse studies and on real contaminated sites throughout LA and throughout Southern California to try to understand what are the environmental and like soil parameters that control this. Like how long does the stabilization last? What happens if the temperature changes or the moisture changes?
Right.
And the reason this is this is important to understand in general, but also important, to support this application, right? Because then farmers could apply these mycorrhizal inoculum to support plants and block metals from getting up.
And they would trust it more, right? When I think that was one of the things
Right.
Taylor Bright another applied mycologist had said, that there was just not enough data to be able to support and have people feel like it was verified and they could go ahead and then invest the resources in doing this.
Yeah, exactly. Oh, that's reminding me of another thing. I maybe later we could talk about. I supported some grade 8 students in this amazing project where they wanted to test whether mushroom fruit bodies take up or accumulate metals if the metals are in
Fruiting body.
the mycelium.
Mhm.
So if the mycelium is growing in a contaminated substrate like a soil or material, like this has been a major question. Like every time I give a workshop, people ask like, oh, but do like the mushrooms that fruit off of remediation experiments,
Contain
like
the
do they
metal.
contain the metals? And it wasn't really known, but it was super cool. These grade 8 students helped figure it out and I helped them analyze mushrooms. So they grew these different mushroom grow kits and they spiked them with different heavy metals. And then we harvested and actually analyzed the mushroom fruit bodies. And so some certain metals were taken up in the fruit bodies and others weren't so much. But
Danielle, I'm going to stop you there for a second. We're going to take
yeah,
a break and then come right back talking about this, the fruiting bodies and the metals.
great.
Hey listeners, quick break here. We hope that you are enjoying EcoJustice Radio and KPFK Los Angeles. You are listening to Renewing the Environment with Mycoremediation with host Carrie Kim and guest Danielle Stevenson of DIY Fungi.
So what did they find, Danielle? Did they find that they were in the fruiting body?
Some mushrooms do accumulate some metals. So some do more than others, like shaggy mane is a known hyper accumulator of toxic metals. Unfortunately, a lot of mushrooms that we like to go foraging for, which are actually fruiting from mycorrhizal fungi, they can accumulate even radioactive metals as well. In this experiment with the grade 8s, they found that certain metals were like, I believe lead and chromium were accumulated by the oyster mushroom. And so I would just say that, there's two pieces for people who go foraging. Always good to be mindful of where you're foraging. Because if there are metals in the soil, like if it was an old mine or something like that, there may well be metals in the mushroom fruit bodies that you're harvesting. But the second piece is that in general, I totally compartmentalize. Like I grow mushrooms. There's so many really cool ways you can grow mushrooms for food on really safe materials like spent coffee grounds or
you know what's going into them because
exactly.
that mystery piece when we're dealing with toxicity.
Yeah. So for remediation, usually mushrooms actually don't form we're working with mycelium. That's the living part of the fungus. But if they do, you just need to incorporate them back into the system or harvest and contain and dispose of them safely. Um,
What does that disposal, safe disposal look like? What would that be?
the basic process is to collect them in a container. It depends. I mean, I've worked on sites that were large enough that we actually had multiple, like totally sealed bins that we would collect into. What I do is I let them anaerobically compost down. So you end up with a bunch of biomass that ends up being a small amount of like sludge.
Mhm.
And then you have a smaller amount of material. You can, if you have access to testing resources, it's best to test it and actually see what the concentration of metals is. Then that will tell you how you have to dispose of it. So if it's above certain concentrations, you do need to pay for it to be treated as hazardous waste at a hazardous waste disposal facility. If not, you can essentially, like, I wouldn't compost it anywhere near where you're growing food or anything like that. You may choose to actually steward that waste on your site by keeping it contained over the long term and committing to stewarding it in that
And then
way.
and then testing it again. It seems like the testing is so essential to really know what's
Exactly.
happening. But even when it's disposed of as hazardous waste, like how is it actually disposed? I don't think people really think that through. I don't know how
I
they
know.
really dispose of
And
hazardous waste. It seems like they probably just put it back in the environment, is what I would guess in some way.
yeah, so I followed this curiosity too. I fully share it. I actually took a job doing a waste audit at a waste facility, and I would just like, it was so interesting to me to actually see what comprises a waste stream and then be able to like follow it and ask like, okay, well now, okay,
Mhm.
I'm handing this styrofoam off. Now what happens with it? And so I
Right.
learned a lot. So it really depends on what it is. Some things continue to get
Pass
basically
the buck.
recycled, like melted or whatever, compressed and then made into another thing,
Mhm.
as long as possible until it's done. Certain hazardous waste materials are very mysterious to me, and when I tried to look into this more, I mean, what I will say is that I mean, this is an eco justice show. I am sure folks are aware of the health disparities and the racial and economic disparities with regards to where these hazardous waste disposal
Sure.
facilities are
Yeah. All the
located, right?
sacrifice zones. Mhm.
Exactly.
Of color.
Disproportionately in communities of color. And so I think, and I think there's a lot of mystery. There's still a lot after digging that I don't know. I mean,
Sure.
sometimes you hear, yeah, again, it depends on the waste, but also sometimes waste, or sorry, hazardous materials are only need to be cleaned up once they're at a certain level, and those levels are often still above the human health risk guidelines. So this is really the crux of what inspires me to do this work and to continue to explore like community based and ecological ways that we can not like truly address and resolve the toxicity that's all around, because there's
Mhm.
so much, like you started the show with, and it's a lot of
It's
contaminated
overwhelming.
sites just sitting there.
Exactly.
And I'll say too, I'd love to touch on testing, and I'd love to just bring connect in with the theme of the show as well, and finish the piece around metals. But part of part of the urgency here is that climate change and projected increases in temperature, atmospheric CO2, increased frequency and severity of natural disasters and extreme weather events like flooding and fires. All of these um, changes are predicted. There's studies that are showing that they are generally actually increasing the mobility of metals and the release of metals and other contaminants. So I saw a study that came out last week about how something like 60% of the Superfund sites in the US are at risk for like
Mm,
flooding and things,
okay.
which,
Okay.
right, the problem with flooding is that it just releases, it spreads
Mhm.
contaminants all over and deposits them when the water goes down. So it's
Because we
really
mhm.
it's really important to, I think, continue to all work together to figure this out, to figure out, again, like community based and ecological approaches to truly resolving the contaminant, the toxic legacy that we're all
It
faced with and
seems
how
that
it's changing.
the toxic load is so great that rather even than trying to scale on a massive level, that this hyper local approach of more and more people engaging in their local areas in
Yes.
remediation is what's needed. We need to be activated everywhere locally. And
Yes.
together that creates a global effect, because
Yes.
it isn't as easy to scale up everything to some, doing millions of acres at a time of remediation.
Exactly, I completely agree, and the other piece is that it isn't always effective, right? Because,
A lot more to
there's
control.
different species here, and they're adapted to this particular environment and
Mhm.
climate and everything. So it's more appropriate, I think, in every way to work locally. You can draw best practices, like general protocols, but then work with local people and local species. And I think there's a real interest always in, I always find this, I teach a lot about mycoremediation and bioremediation, and I think people really get excited about like going somewhere far
Right.
away to like, oh, I want to learn about that project and like
Mhm.
learn about, yeah,
Right.
remediation in like the
Restoration.
jungle or something. And then I'm like, well, hey, I always just encourage people to
Do it in your
like look
backyard.
around, like for sure where people live, right in someone's neighborhood. There's contaminated sites, there's toxic waste, you
So
know,
much opportunity,
there's
yeah.
so much to do, and I think we really would all benefit from that. Like folks putting energy in locally, and developing local solutions and
And
seeing
local
what
networks.
works.
Mhm.
Exactly, yes.
What is your work? Can you speak a little about your work with healing city soils?
I'd love to. Yeah, actually that perfectly ties into to testing. So when I started doing this work many years ago, I quickly ran into the barrier of of just testing. It's really expensive to
Mhm.
do proper testing, and it's also very important. You need to know what you're starting with and then if it's changing with your treatments, right?
Mhm.
And so
Mhm.
it's super, like, just to give folks an idea, like one sample analyzed for metals and just general hydrocarbons can be up to $400 for one sample.
Yeah.
And
Mhm.
that's not so.
Yeah.
I started Healing City Soils to make soil testing accessible and free to people. And
Free.
so,
That's amazing.
yeah, so I wrote a letter actually to my local paper and proposed this project, and I was contacted by professors from local universities and colleges, and they said, hey, this could be a real win-win. We have environmental science students that need to do
Yeah.
projects and get experience with this kind of thing. And I partnered with a local compost education center, university, and basically students take soil samples, and they're analyzed for metals, and the results get sent back in personalized memos with free fact sheets and workshops on, okay, what do
Mm.
your results mean, and then what do you do about it. And so it's because that's another barrier, is that sometimes folks I mean, I know for me, when I first got test results back, I was like, what does this mean? I
Right.
don't
What to
know.
do with it now? Yeah.
So, and so I'm really excited. So this program has been running for 6 or more years now. And now we're actually launching a bioremediation component. That's always been the goal. So because once, like that information on, well, what what next, what do we do, what can people and communities
Once they find what's in their soil.
do exactly. And so we're actually doing a pilot study right now in partnership with a number of First Nations and different amazing community groups. And we're piloting out approaches working with native plants and native mycorrhizal fungi and compost. So really simple, accessible treatments. And we're going to see how that works, and then provide, again, fact sheets, information at the end of the study on how it worked, so that
Is
other
that
folks
locally
can
in
replicate it.
Is that locally in Southern California, First Nations from Southern California, or is it throughout, or where the
No, this is actually in Canada. So this is up in Victoria and Lekwungen speaking territory. And so, but I will say that since I moved down here, it's been an honor to just meet and learn about the amazing work that Orange County Environmental Justice is doing regarding soil lead. And so, yeah, there is some really amazing work happening locally. And part of my research actually through UCR Environmental Toxicology program is I mentioned that I'm going to be doing remediation on real contaminated sites. And so one of my partners is Orange County Environmental Justice on one of the sites they've identified as contaminated in Santana. And then I have a number of other partners, LA Eco Village in LA, and a few other partners. So I'm really excited and honored, because again, this piece of
Just
local, like figuring out next steps locally
right.
for local communities who are engaged with the land and engaged with and have encountered soil contamination. Like, okay, what next? Well, so I'm really excited to be able to work on this with these amazing partners locally in
Right,
Southern California.
just like with farming, organic farming, regenerative farming, you need this massive population of people to start doing this.
Yeah,
Whether it's also the research, just the research alone and the testing alone, all those things, all those pieces need to come into place.
exactly. And
Further the field.
I'll mention too, for people who may be listening and wanting to connect to testing, I'll say that universities are a good place. If you can find a student, someone like me, if you're local, reach out to me. So, universities often, there can be some shared goals. I mean, university labs often have the analytical equipment to do contaminant testing. But then another option too, that's accessible to the sort of community member is bioassays. So there's ways that it's not the same as you know a soil test, but
It'll give you some idea.
bioassays give you some idea, at least, like, well, how contaminated is this soil?
Mhm.
Like, how toxic is it to life? And then also, are we making an improvement? Are we reducing the toxicity with what we're doing? And
It
so,
seems
yeah,
incredibly important because, with people, especially since the pandemic and more people turning to farming, whether that's urban farming, and people really need to be educated about, well, your soil may not necessarily be perfectly healthy at this moment, and it likely is not.
yeah,
There's a great potential of things like lead and, as we know, heavy metals, whatever might be in that soil, and then they're consuming food produced from that soil.
yeah, exactly. And so there's really simple things people can do to prevent that. Even if you can't get a soil test, you could probably assume, like, that there's lead from the air, from the soil that's been building up. And so you definitely want to, I mean, to be really safe, you could grow and raise beds. It's not always accessible to everyone and not on a larger farm, but at the least, make sure that you are mulching lots of organic matter.
Mhm.
Often lead is actually in in the dust. And so you get exposed not only from eating the food, the veggies, but also from just the dust that's on,
Right.
you're exposed to. And so washing your produce, I mean, it's a bummer. I know my grandparents, like, there's something just beautiful about eating like a dirty carrot right out of the
course.
soil, like
Yes.
our grandparents did,
Yeah.
but
Especially when
I
the soil
think
is healthy.
when the soil was healthy, right? And so I think I'll just say for now, there's lots of resources online people can look up about safe, like metal safe gardening and food growing. But
Oh,
yeah, at least wash your produce, mulch your garden. If you think you might have heavy contamination, just avoid growing root crops. Usually any kind of fruit is fine, like
mhm.
tomatoes or things like that. So yeah, there's some simple things people can do. But again,
What do
yeah,
you recommend for mulching yourself? I mean, and those are all various things that people consider as mulch, but for you, when you say that, is
yeah,
there anything specific you have in mind?
I
Material you have in mind?
usually I usually again, I look to the waste stream, and so you can sometimes cities or will have programs where you can actually pick up, you know, leaf mulch and things like that for free. I mean, since I've been here, I definitely look at those palm fronds that are always blowing down, and I haven't played with that too much as a
Right.
mulch, but just any kind of organic material that you can be pretty sure is clean, like hasn't wouldn't have a some kind of contaminant.
Not next to a
um
bus stop.
exactly, yeah. So, but I mean, it can be hard. It can be hard in a big city to connect with mulch. I mean, I can also sometimes, if I can't find any organic material for free that's just from the waste stream, you can even just buy a bale of straw, and that's a really great mulch.
Yeah, we tend to use a lot of our organic alfalfa ourselves. But, Taylor, we're
Yeah.
coming to the end of our time, surprisingly enough. It just went so fast. But I would
Wow.
love for you to give your information so people can connect with you, especially if listeners have people that would like to have their soil tested and just to maybe be involved in volunteering for some of your projects. Just let us know some of your contact info.
I'd love to. Please do reach out. If you'd like to learn more, come to a workshop, help out with some of these remediation projects, or just connect and have other ideas. You can reach me by email. It's Danielle Stevenson at gmail.com. And you can find me DIYfungi.blog is my mycological blog. I'm on Instagram and Facebook too under that name, DIY Fungi.
Thank you, Danielle, so much for sharing all of your passion for the fungi and for restoring the environment. I hope that many, many people are inspired and galvanized to continue this work with you.
It's my pleasure. Yeah, please join. Let's make it happen in Southern California.
Absolutely. Thank you again.
Thank you to our guest today, Danielle Stevenson, and thank you to our listeners for joining us. This has been Renewing the Environment with Mycoremediation. Please connect with us on social media at EcoJustice Radio and SoCal 350. If you like what you heard and you want others to be informed, subscribe to our podcast, share the episodes, and help us continue our efforts by donating to the show at ecojusticeradio.org. You've been listening to EcoJustice Radio on KPFK Los Angeles, a project of SoCal 350. The show can be found on kpfk.org, all major podcast apps, and at ecojusticeradio.org. Created by Mark and JP Morse, executive producer Jack Eidt, producer Jessica Aldridge, engineer Blake Lampkin, interview hosted by Carrie Kim, and original music by Javier Cadre. And until next time, remember, the power is yours.